Her sort of research started when one of her children, I think, had some toxins within and was exhibiting symptoms, and she sort of delved into this side of things and how your food and what you're eating and what goes into you really makes a difference, and was able to heal her child. So I asked her to come, and she suggested talking on something a little bit more sort of research and scientific based, but I think it's good to have a sort of difference and a wide variety of different things being talked about. So welcome, Katherine, and thank you for coming.
Thank you. Well, wonderful, wonderful. So my name is Katherine Reid, and I'm the executive director of Unblind My Mind, which is a nonprofit focused on, really thinking about improving our health through foods. I am a biochemist, and I love science. I am such a geek. So, if just give me a sign if I'm like drowning you in science, and I'll kind of try to bring it back up a little bit, 10,000 foot higher. We'll go in 10,000 foot increments. So thank you very much for having me here and listening. Unblind My Mind, like I said, the whole mission of our mission is to really unblind and raise awareness of the links between the foods we eat and the chronic illnesses we suffer. It definitely was as a result of our personal journey through helping my daughter, which I'll mention a little bit in here. First and foremost is like educating people and really kind of giving them the information and thinking about some of the science that supports, what we're doing here. But another big element is training. It's enough, here's the information, here's the science. But when you actually ask people to redefine food and change their lifestyle, it's not just something that is easily adopted. And so, kind of bringing people through that and having a community that supports them, such as conferences like this, is an important part of the training to get people actually incorporating a lifestyle for life. And the reason that this was established was not only was I seeing just incredible changes within, my immediate family, my daughter, myself, and then extended families, and then I was extending it to friends, and I was like, wow, if you look at the statistics, we need a change in how we think about food. Over 25% of Americans suffer from a diagnosable disorder affecting the brain. And notice, I'm not saying mental illness. We really have to change the way we think about mental illness. It's, issues that are affecting the brain. With 18% suffering anxiety, 9.5% suffering depression, over 1%, afflicted with autism. And disorders that impact the brain are now the leading cause of disability in the United States and Canada. Now, outside the brain, we then have all these metabolic issues where we have 2 out of 3 adults that are considered overweight or obese, and 9.3% of the U.S. population has diabetes. So, clearly, we need to be thinking about what are we doing in our life, and what control do we have? And it turns out we have a lot of control. So today, I'm going to really focus on the microbiome. And what I love about this picture is this was, I put this slide together when we were talking about the 99% in an economic sort of way. But it's like, we have to think about the 99% of the genetics within our GI tract. These are the microbes of bacteria, viruses, archaea, and the fungal that populate our GI tract. And our GI tract is the densest ecosystem in the world. I mean, that's just a profound concept, that 99% of our genetic makeup are these microbes, and the densest ecosystem in the world is the human GI tract. So we're going to talk a lot about this, and there's a sub-theme here about not only reducing toxins, but also thinking about the inter-kingdom communication of these microbes and what they're secreting out of their metabolism, and how does that affect us as human beings. If you think about our whole neural network, I'm moving this, I'm deciding, I've got conscious movements, the neural network is really very much influenced by these microbes within our GI tract, and that is just fascinating to think about. So when we think about the influence of these trillions of microbes within our GI tract, the largest influence is the 50 tons of food we choose to eat in a lifetime. Think about that. It's like the weight of that mother whale. 50 tons of food that we choose to eat. So our choices are absolutely influencing all of the microbial populations, and we are able to control 99% of our genetic makeup through the foods that we eat. Now, just like any ecosystem, it's very vulnerable to imbalances. You think about this pond that's overrun with algae, where that algae is now depriving the rest of the ecosystem of vital nutrients and being a balanced, well-thriving ecosystem. Conversely, we can have an ecosystem where we have harsh chemicals or harsh environments where there is absolutely no growth, lack of abundance, because the environment is so hostile. These imbalances absolutely occur in the GI system and are the leading cause of many, many chronic illnesses. So we're going to be talking about the two important things here is diversity and abundance. And those are huge protectants of, being able to ward off some bad actors or invaders that come in. And how do we achieve that? And so just here's a little depiction of where we have, microflora, and all of these endotoxins and metabolites that these microflora are producing can influence all of the the digestive tract inflammation from IBD, IBS, or CRC, metabolic diseases like diabetes, genetic diseases, liver diseases. They don't have heart here, but it also influences the brain. And there's all sorts of now scientific support that the microflora are driving a lot of these inflammatory sorts of triggers that infects the entire system. So I'm going to talk about 3 relationships that we have with our microflora. Ideally, we want a mutual relationship where the microbes are actually producing metabolites and nutrients that are very well helping the human host thrive. Okay, that is a mutualistic. The microbes are benefiting, the human is benefiting, and that is a mutual relationship, and that is what we are striving for. We often hear commensal, but commensal is not the optimal. Commensal means the microbes are surviving, and it's not making us thrive necessarily, but we're surviving. But we want thriving. So thriving and surviving, I'm going to make a distinction. And the commensal is not necessarily these microbes that are helping human health. They're just not whacking us down, and we're kind of, chugging along through life and getting through. The worst relationship is a pathogenic one, where now these microbes are actually deteriorating our health and our survival is now impacted. This is the relationship, obviously, we want to ward off at all costs. And the more that we are protecting ourselves with, again, abundance and diversity, the less vulnerable we are to these pathogenic invaders. A lot of my research recently has been focusing on, how does the microbiome impact behaviors? And we think about, every behavior that, humans have from, okay, I'm anxious or I'm depressed, or it starts to you start to look at the influence of the microbiome coming from diet and stress and environmental toxins and probiotics and antibiotics, and you think about, okay, how is that impacting behavior? And we think about it as going through the brain, but a lot of it is about just impacting the whole nervous system in general, which, of course, the brain is part of the central nervous system, but that's not the only nervous system. It's really impacting the entire neural structure. So when I think about behavior and I see somebody who's not acting necessarily happy or optimal, and I'll give you an example of my husband, because I can always put him under the bus. If he comes in with a look on his face, and now, being on this journey for 8 years, if he comes in and walks through the door, I'm like, whoa, what did you eat? I can just see it on his face. It's just like this, weight and, depression and kind of anxious or sometimes irritable. And I'm like, what'd you eat? And almost inevitably, he has eaten something at lunch, as a business lunch or something, and he's like, yeah, I'm not feeling so good. I ate that salad dressing. I thought it was going to be okay. And that's all it takes is like a little bit of salad dressing that, had lots of additives to them. And I can see lots of times, even with myself, my child, my first question is, what'd you eat? So it's looking at mental health in a whole new way. So what I like to focus on here is thinking about we eat foods, and then you go through the digestive tract, and these microbes go through fermentation, right? They're fermenting or metabolizing the foods we eat. Of course, we also have human metabolism, and the metabolites are the products of that metabolism. So eat food, humans will have a metabolism, but not all foods are actually utilized by human cells. In fact, a lot of them we have to think about when we eat foods, we're also feeding the trillions of microbes. What we eat is not just about human metabolism, and I think lots of older studies on nutrition were flawed because they were thinking about it from a point of human cellular metabolism, but they weren't considering the trillions of microbes and their metabolism and what they're producing. So if you think about the ecosystem picture, we're thinking about when we eat these foods, these microbes produce these chemicals, and if those are in excess, those might be driving a certain inflammatory response or a good response, and we have to think about the balance of the chemicals, not from just humans. And I like to think of it as a subway map. Now, the intent is not for you to be able to see every single metabolite on this picture, but that we've got a map of all of these different metabolic cycles, and here is the human metabolic cycles and how they intertwine and interlink. But what's fascinating is if you look at microbial metabolism, let's say bacteria, a lot of the bacteria metabolism will use and hijack similar sorts of metabolic cycles in the humans. And so you have an intertwining where you have one metabolite that might be secreted from the human cell that's being then picked up or utilized by a microbial cell, and you get this intertwining of metabolism that is very much linked. And you get this communication going between them. Oh, if you have an excess of this metabolite, you might have a human cell function actually stop because it's not getting what it needs because it's just been hijacked and pulled away by a bacterial population, a fungal population, and so forth. So from a scientific perspective, I love to think about the intertwining and the interkingdom communication between these microbes and humans. The first one I started, and Christine was mentioning that I spoke here last year, I talked about glutamate. And I talked about glutamate and how it's coming from the food supply, how it's being enriched in the food supply, and how this is hijacking our nervous system. And that's all true. And you can go on unblindmymind.org, and there's some media, and even the last year's talk will talk about glutamate coming from the food. But now I'm going to take a different spin here and think about glutamate coming from microbial fermentation and how we can actually make sure that we're controlling that part of the glutamate, because we can control the food a lot. But what people aren't thinking about is how is that coming from microbial populations. So glutamate is an amino acid. That amino acid is the building block of proteins. That's how proteins are made. And each subunit in a protein is called an amino acid. So glutamate is an amino acid that is metabolized by every single organism on Earth. Every single little organism out there, from plants to little bacteria to mammoths, can metabolize glutamate. It's an excitatory neurotransmitter in humans that's responsible for exciting, meaning initiating a neural signal, in 40% of our nervous system. It's used in all sorts of functions. It's almost like the ubiquitous chemical that is allowing us to go and communicate from neural signaling to inflammatory signaling to immune signaling. It's used in energy balance, digestion regulation, ammonia balance, memory, learning, inflammatory and immunological responses, and so much more. You're going to hear me talk about glutamate signaling. And this is how glutamate actually is so ubiquitous, or so diverse, in its functions, is we have these glutamate receptors all over our body on the cell surfaces, on our skin, on our organs like the liver, the pancreas, the heart, and they have these cell receptors called glutamate receptors. Glutamate binds to those receptors and activates a certain function. So what we have depicted here is what happens with two nerve cells and how they communicate by using glutamate to propagate a signal through the nervous system. Similarly, you can have that happening with, okay, we're going to be releasing insulin. That's regulated by glutamate receptors binding onto these cells and releasing insulin, or glucose regulation. So you've got essentially one key that's unlocking many doors with an incredibly diverse set of functions. So that's all fine and good, but we have to make sure that we keep this regulated and we don't have a bunch of excess sources coming in. Fortunately for you, the sound's not on. This is my daughter screaming, actually, so it's very profound. Okay, so it sounds like there's not a sound cable on there, and like I said, it's probably for your best. This is my youngest daughter, Brooke, soon after she was diagnosed with autism. She's here and she's screaming, and I actually was videotaping this to send to a neurologist, like, what in the heck is this behavior? And in this particular video, she was trying to decide between, having a movie on and having a movie off. And it was like this perpetual level of indecision that was like this neurological loop. And as a scientist, that's what it looked like to me. It looked like she was stuck. It reminded me of a seizure-like disorder that was manifesting itself with a physical manifestation and behavior. I couldn't pull her out. This particular episode lasted for 4 hours that ended with her complete exhaustion. She just collapsed, and then when she woke up from her nap, she resumed the exact same loop. It was like that neuron fired again, and because it was so strengthened with every single turn of the cycle, it was becoming stronger and stronger in part of her neural mapping. As a scientist, I was like, okay, I have got to figure this out. And of course, as my daughter, I'm like, I have got to heal my child. And so I started to dive into what could possibly explain such a behavior that, is obviously becoming more and more common in our population and with our children. And so I started to really probe the gut-brain connection and thinking about how is it that the gut is communicating to the neural structures that could potentially result in such a behavior. And this is what started me along the glutamate pathway, is that when I looked at glutamate and autism and glutamate and other sorts of things, I found that that was a very common theme with a lot of chronic illnesses is glutamate, and I'll get a list there in a minute there. And so this is how my journey started in thinking about microbial metabolism, foods, and how these microbes are hijacking our system, essentially, and driving our behaviors. On a very basic level, glutamate can look like this, and you often don't hear about all of the metabolic cycles that glutamate's involved in. You'll hear about glutamine, and that's often being recommended as a supplement for leaky gut. But I often see people who are reacting to glutamine because glutamine can be converted to glutamate very readily. And so you get this excitatory signal that people sometimes are experiencing with these glutamine supplements. Glutamate drives GABA, which is the inhibitory, the brakes on the nervous system. And you want a nice balance between glutamate and GABA. But we don't have a lot of GABA-fortified foods, and we don't, and there are microbes that help produce GABA. But what we have is a much more tendency for the glutamate to be excitatory, and that balance gets tipped often with our stress, with our environment, with our foods, and our microbial populations. And glutamate can also go into the Krebs cycle, or the energy cycle, which is located in the mitochondria, called alpha-ketoglutarate. And so this is how glutamate can then actually drive energy cycles. So you've got this, and this is not even fair to say it looks that simple. It's actually here. Glutamate is involved in all of these cycles, which is why it's metabolized and utilized by every single organism on Earth. You have it balancing energy, which is a carbon cycle over here, But it's also part of nitrogen metabolism and balancing nitrogen. And you think about nitrogen balance in plants and so forth, and in every single mammal, and you've got histidine, and you've got urea, and you've got all of these pathways that are actually very much including and driven by glutamate balance. And so glutamate is very important to keep in balance. So when I started to investigate glutamate and thinking about maybe the root causes of what was going on with my child, there was no lack of literature to support such a hypothesis. There's over 1 million scientific publications of various diseases and disorders that are associated with glutamate dysfunction. So I kind of felt like I started to hit the mother lode. We weren't just talking about autism. We're talking about a lot of new age modern diseases, from diabetes to obesity to Parkinson's, schizophrenia, that are absolutely a glutamate dysfunction issue. A lot of the research the research is being funded by pharmaceutical industries looking at glutamate blocker drugs. They are a huge blockbuster industry where you're trying to block the glutamate from binding to the glutamate receptors. But as I said, glutamate's involved in so many different functions. You start getting glutamate blocker drugs, you are not able to control that it's only blocking this one particular region. You are going to have side effects, and that's been the biggest issue of trying to control these glutamate blocker drugs, is it comes with all sorts of side effects. So then it becomes this daisy chain of prescription drugs of trying to then, oh, well, you've got that symptom, let's block that out. And so when I started to see the scientific literature, and like, I can't go through the glutamate blocker drug approach, I started to look at all the ways that I could control glutamate in what I did the experiment on myself too, and my daughter. And I did it through foods and thinking about the microbial populations. So like I said, I talked about foods quite a bit in the last talk, and so what I'm going to focus on today is the microflora concept. So, foods are obviously driving the microbiome and its metabolites, but, for more information, you can go on the website for the food. And the microflora and the inflammation, are also closely linked. Inflammation creates a very different environment in our bodies. And as a result, you're creating a different environment for the microbes, particularly if this inflammation is occurring in the GI tract. There are some microbes that thrive in that inflammatory environment. It's got lower pH, it's got a completely different environment, you've got different metabolites and so forth, the cytokines and things that are being secreted that make it a different environment. So we actually have quite a bit of evidence that shows that there are microbes that love and survive and thrive on glutamate. And that's not good, because then they're looking for all these sources and ways of trying to increase glutamate within the GI tract and beyond. So it becomes an osmoprotectant. And as an example, bacteria such as E. coli will actually synthesize glutamate to be able to promote a high osmolality so that it can thrive in its environment. And so you've got something that's actually directly secreting glutamate because it helps it thrive. Excuse me. The Borrelia bacteria associated with Lyme disease can't synthesize glutamate. But we all said that every single organism needs it. So it will actually upregulate glutamate transports and scavenges it from its host. That makes it more virulent. And we all know what happens when that Borrelia bacteria becomes more virulent, the Lyme and the symptoms associated with that. Only somebody who's gone through that can really describe that in detail. It's awful. And so you have these, people who don't know how to use the laser. So we have these, two cases right here. And then, because, like I just said, inflammation usually results in lower pH, and there's oxidative stress conditions, you've got microbes that just love and thrive in that because there's less competition. If they can survive in that harsher condition where these microbes can't, they have a survival benefit. And so what they do is they use these glutamate shunt pathways where they bring in glutamate that's increased in an inflammatory environment. By necessity, that's exactly how our immune system is communicating with the nervous system. So it's able to shuttle glutamate through, keep the pH the same in its own cell, and be able to survive in a low pH environment. So those are some examples, and there's so many examples, of various bacteria or microbes that are able to thrive in an environment that is not necessarily beneficial to the host. And so you start to get a vicious cycle here where these microbes actually might even activate inflammation, because when our cells are inflamed, our immune cells, they secrete glutamate to signal stress. Stress, help me out, body. I need some actors to help with the cytokines in this thing. And so in doing so, they're secreting glutamate, and if the microbes like that, they might now have a survival benefit with increasing inflammation. That starts to result in this glutamate buildup that they are thriving on, and that starts to drive this imbalance, where you're starting to then have an environment more and more that is supporting a different unhealthy environment rather than the one that we seek, and you get into this state of chronic inflammation, and it's this vicious cycle. So that's one example here of one metabolite, and we've got thousands, of glutamate, and how the microbiome is absolutely influenced by that environment with glutamate. Additionally, when you think about the GI tract, we're thinking about, again, the interkingdom communication. And there's been a lot of research that's shown that the enteric glial cell, they're like the little environmental patrol, agents in our GI tract. They report on the environment to the brain and the nervous system. So they're strolling along here, but they're being trafficked by the microbes, mainly through glutamatergic signaling, through glutamate signaling, and they are responding to the microbial metabolites that are driving them to the mucosal wall to respond to the environment. So if that's a mutual sort of relationship, they're communicating, hey, everything's okay, and we're reporting back, and there's no inflammatory response. If these microbes are kind of signaling, if these microbes are starting to signal, hey, we are having a ball here, and the enteric glial cells are picking up an excess of metabolite that is not healthy to the human, they're then signaling the stress response, and you're getting a very different response getting reported to the control unit of the brain, and the brain is then responding back with inflammation to the GI tract. This is how the mucosal integrity starts to get impaired. It's starting to subject these glial cells to more and more of that toxic metabolites, and they're very much important for the integrity of the cell wall. And you start to get these poor little glial cells that are being subjected and bombarded to the excessive metabolites, and then you start to get a little bit more holes and permeability through the GI tract that then starts to cause a whole host of toxins going into the system. So these enteric glial cells and the signaling that they receive influences all sorts of motility function, immune and inflammatory responses, and barrier integrity. And so just kind of as a last note on the glutamate thing, and we're going to move on to other examples of metabolites, you get these microbes that are absolutely addicted to MSG just like the humans are. And so why am I saying MSG all of a sudden? It's monosodium glutamate, and glutamate have no difference in terms of biological activity in our bodies. So we often hear about the common name of MSG in our food, but it's really glutamate, the free glutamate. That's the active component that is the metabolizing and signaling and activating our nervous and immune system. And so here's some literature on glutamate in support of the mood disorders. This one just came out in 2018. Couldn't support what I'm saying more. It's talking about how the sensory cells are linking up to the nervous system and signaling, and that's through serotonin and glutamate. And so you couldn't get a more direct gut-brain linkage in how this is impacting moods than that recent article here. And of course, that article 2018 is built up from a lot of other support literature. Glutamate-based depression. A lot of people don't realize how much depression is a glutamate sort of signaling function. And then there's more. So I'm going to talk about now one more step up, and where do we get excessive glutamate coming from our food supply? And so I'm going to talk quite a bit about protein fermentation. So we all need protein as a nutrient for energy, and it's all about the quantities that we consume in the diet and what we're consuming that protein with. An excess of protein fermentation can result in glutamate as being a byproduct. And that is a huge source in the GI tract for glutamate production that's not coming from food, is when you start to have your microbes dependent on fermenting protein, and it's coming up with glutamate as a byproduct. These microbes are extremely adaptable. We know what their preferred food is based on our preferred health state, and I'll get into that towards the end. But if microbes can survive in an oil spill and do honky-dory, they are adaptable. They will absolutely utilize whatever they can for their survival and can adapt. And they have these genes that will actually switch on and off depending on their environment. And so if their environment in the GI tract is a lot of proteins without the fibers, and we'll talk about that, they'll start to ferment those proteins, and the byproducts associated with protein fermentation are so linked to all the inflammation issues that are going on in the GI tract. So they've got proteolytic power here, meaning they've got proteases that takes this protein and converts it into free amino acids, glutamate being one of them. Additionally, we have a whole host of other things that you might hear are in excess in people's bodies. You know, it's not traditionally measured at a doctor's office or like that, but you can sometimes actually smell these things, you know, and actually the smell and some of these symptoms can be very big clues in terms of, okay, I probably have an excess of that going on. For example, ammonia. You know, people will say, okay, I'm starting to really smell an excess of ammonia going on. Now, certainly that can mean that there's something going on with the kidney, but that typically means there's excessive amounts of protein and fermentation going on where you have a lot of ammonia production. Hydrogen sulfide. Hydrogen sulfide we actually need. It's an important neurosignal, but again, in excess, it's extremely toxic. And so hydrogen sulfide and the sulfur smell with, stool elimination is a classic sign that you've got too much of particular bacteria that are producing this hydrogen sulfide. What else is another one? Phenols. I often hear about people coming to me about phenol sensitivity and, okay, I'm going to cut out the polyphenols in my diet because I'm phenol sensitive. But the main driver of where those phenols are coming from is protein fermentation. Clostridia bacteria love to ferment protein, and their byproduct is phenol. And I'll go into that example of why they do that sort of nasty sort of thing. Let's see. And then keto acids are another one. Again, you see sometimes imbalances in those, and it starts to hijack the energy cycle. And then short-chain fatty acids and branched-chain fatty acids we'll talk about more, because that's sometimes what we're kind of looking for to nourish our colon cells, and how do we get more of those. So protein fermentation can produce quite a bit of chemicals here. So phenols, working with a lot of people in the autism community, I hear a lot about phenols and phenol sensitivities. And, if you think about the Feingold diet, it was initially about, the phenols and mostly dyes. But a lot of the phenol sensitivity starts to come from the Clostridia, and they produce that from tyrosine and phenylalanine. Tyrosine and phenylalanine are what's called aromatic amino acids that are very concentrated in animal proteins in particular. And Clostridia thrive on this. They produce phenols for their survival benefit. Phenol is toxic to a lot of other bacteria and organisms, and by them producing that, they've pretty much carved out their little territory. But phenols for humans requires it to be detoxified in order to be eliminated. So we have to then use our detoxification pathways like sulfation to take that phenol and get it out of our system. So you can imagine that the more toxins that you have being produced in your gut, the more you're taxing all your detoxification pathways that you should probably have to, help and protect for other things, rather than this slew that's coming off in the microbial metabolism. This particular chemical here, 3-hydroxyphenyl, 3-hydroxypropionic acid, which thank goodness is abbreviated HPHPA, I often see when I'm looking at metabolic profiles, that's elevated. And that will drive a certain imbalance between dopamine and neuroepinephrine. Again, there's another link as to how is this inter-kingdom communication affecting our behavior. You get elevated dopamine, low epinephrine and neuroepinephrine, you have a classic behavior trait associated with that imbalance. P-cresol is often associated with Clostridia difficile. Again, it is very toxic and requires similar sorts of pathways, and it too will interfere with this neural balance. Tryptophan is another aromatic amino acid that will be resulting from protein fermentation. Now, tryptophan has an incredible benefit that, if metabolized and utilized in the proper way, is actually producing serotonin. And serotonin is, again, lots of cell receptors that contain serotonin, and we hear about serotonin uptake inhibitors because people are producing too little of serotonin to be having mood regulation and getting enough serotonin to the brain. But what most people don't know is 90% of the serotonin is actually produced in the gut, a lot of that influenced by the microbiota populations. But the other thing that's tricky is that when you have tryptophan, you can't tell it where to go. You can't say, tryptophan, we want you to go to serotonin, produce serotonin, and then serotonin is necessary to produce melatonin, and that's good for my sleep cycle, so please go on this pathway. You can't tell it to do that. And inflammatory sorts of pathways are unfortunately also triggered by tryptophan. So tryptophan's actually, in an inflammatory sort of condition where there's stress environments, it's producing quinolinic acid, kynurenine, prostaglandin signaling, which are all the inflammatory sorts of pathways. So you then have tryptophan that could be beneficial now turning into this chronic inflammatory signaling, trickle. The less that the tryptophan is received in a non-inflamed environment and the microbes are happy and they're not in the stress signal and you've got, a good environment, you can be more assured that the tryptophan is producing the serotonin. Again, it's environmental sensitive. So other neurotransmitters, I kind of just touched on a little bit of these, is the GABA, dopamine, neuroepinephrine. These are all neurotransmitters that are coming from protein fermentation and the microbes that are, depending on the microbe populations. And so you've got a lot of examples here where neurotransmitter balance is absolutely controlled and regulated by the microbiome within our gut. Cyclic dipeptides, a lot of people don't really hear much about that. Dipeptides are just two amino acids linked together. These are signaling molecules, too, that are also another protein fermentation product. They are also being snuck into the food supply like you wouldn't believe, and people are just not aware of that. And so, again, the more that we avoid processed foods and ingredients that we can't recognize, the more we protect ourselves. Hydrolyzed collagen, and I did hear that in the previous talk, it's a big source of all of these cyclic dipeptides and signaling molecules and protein fermentation, which often people do not have balanced. So I'm always cautious about the gelatin and collagen being added to the diet. The cyclic dipeptides are signaling molecules not only used by us, but also the bacteria. It's like a quorum sensing, meaning they're throwing it out there to say, okay, hey, we've got a good community down here and we need some more people, come on down here and have a party with us. And these are communicating population size and regulating symbiosis within their little ecosystem that are all able to, pick up this dipeptide signaling. And it also then will communicate when we become virulent to the host. Okay, hey, attack, boom, and they act like a major organ. We think of them as like these little suckers that are unicellular. But when you add trillions of them, they act like an organ when they're quorum sensing communication, and now they can move into big, huge destruction masses when they have the capability of being able to communicate with themselves. to do a certain function that's maybe beneficial to the human host, but it also could be extremely deleterious. Mammals also use the cyclic dipeptides to regulate neurological activities and mediate inflammatory responses. And it's this interkingdom communication where if the bacteria know that, oh, hey, we can produce some of this cyclic dipeptide stuff and we're going to get this inflammatory process here, and that helps us to survive because we're creating a hostile environment, you've got a non-beneficial interkingdom communication going on here. This one I kind of love here, but I got my little picture of a cell, and it's all exciting here. You hear a lot about mitochondrial dysfunction, right? And the mitochondria, as was mentioned yesterday in the dinner by Jeffrey, is the powerhouse of the cell. The mitochondria are these little suckers in here. So you've got the outer cell and the cytoplasm area here. And the number of mitochondria within a cell depends on usually the cell type. So cells in the liver or the heart or the muscles often have many different copies of the mitochondria in there because they're trying to produce energy for the cell. And it's a high oxygen power need for the cell. So the mitochondria, their structure closely resembles that of the bacteria. And so a lot of the chemicals that the bacteria will secrete actually are able to get into the mitochondria, and I'm not even talking about mitochondrial DNA swapping. That's a whole other talk. I'm just talking about the chemicals and metabolism that they're doing to able to hijack the mitochondrial cycle. They can get in there and they can start to utilize the exact same metabolites that they use for their energy cycles. And so mitochondria dysfunction typically is what I like to call an energy utilization issue. The energy that we are getting from our foods is not efficient or optimal because we have some interference going on through the microbiota. Excuse me. And so here we got the structures. So the gut microbiota can regulate, host immune responses and energetic metabolism, and there is a correlation between the microbiota quality and diversity with mitochondrial function. And so that's a huge leap here. And this, this crosstalk, this guy here, Yates Shamol, microbiota mitochondrial intertalk, fascinating about how we're getting cross communication where they can directly hijack the energy of the human host cell. So here's some examples here. The E. coli and Salmonella can produce a large quantity of the hydrogen sulfide. Again, there's that chemical. And that will start to degrade, it's due to the degradation of the sulfur amino acids. And this inhibits cytochrome C oxidase, which is the mitochondrial respiratory chain. That's exactly what you need for the electron transport chain. It's getting hijacked because it's inhibiting this particular enzyme. Nitric oxide impairs energy metabolism by blocking or reducing this acetyl-CoA. And acetyl-CoA is exactly what needs to go into the mitochondria to drive energy production. Another one is it can alter reactive oxidative species. These reactive oxidative species are actually part of our signaling to sit there and say, okay, we need help, we need an inflammatory response, we need an immune response. And if the microbes suppress that, they have a higher chance of surviving. And so you've got these, nitric oxides, hydrogen sulfide, short-chain fatty acids that are kind of going from the microbes into the cell. And a lot of these are going right into the mitochondria and interfering with the mitochondrial cell function. Similarly, we want some of these things to produce cytokines, and that's some of the synergistic or beneficial things of the short-chain fatty acids that might come in here to try to help induce an immune response to ward off any imbalance that's detected by the humans. So reactive oxidative species and mitochondrial messaging, these are key signaling components of immune and inflammation response. It's released from the mitochondria, right, these reactive oxidative species, and directly correlated to the activity of the electron transport chain. When I see these metabolic profiles, there are so many people that have this electron transport chain completely just shut off. and it's this reactive oxidative species that's being interfered with here. Depending on the concentration of the reactive oxidative species, the function is to induce cell proliferation or differentiation, cytokines. The cytokines are the chemical signals that help us, regulate our inflammatory responses or immune responses, or cell death by apoptosis. So it's a critical way to say, okay, I'm getting this concentration of reactive oxidative species, abort, kill, boom, I'm going to blow up the cell because I just realized that I have a major pathogen in me, I'm aborting. So this is important for how our body wards off infections, and that's all driven by the concentration of that reactive oxidative species. So when the bacteria reduce this mitochondrial reactive oxidative species production, it's for their survival because it's blocking the immune response or the cellular response to that particular microbe. And here's their survival benefit. They increase the superoxide dismutase, which again helps lower the reactive oxidative species and reduces the host cell apoptosis. They want the cell just chugging along, providing them nutrients. So that's another example there. So now I'm switching to a different type of metabolite called sugar alcohols. We don't often think about alcohol coming from anything but our drink of wine or our bourbon or whatever your taste preference might be. But I'll look at these 3-year-old, 5-year-old, 7-year-old metabolic profiles, and it looks like somebody who is an alcoholic. Their cellular metabolism has completely been altered by the production of alcohol coming from fermentation in their gut from the simple carbohydrates, the fruits, certainly the refined sugars, that's producing an abundant amount of these sugar alcohols, glycerol, ribitol, rhabitol, xylitol, sorbitol, mannitol, ethanol, doesn't matter, it's an all. And the pathway for metabolizing alcohol, regardless of where these sources came from, is the sugar alcohol plus this cofactor here that you see is responsible and necessary for so many metabolic processes. This nicotine amide adenine dinucleotide is in the protonated state here, whereas at the other side of the reaction, it's reduced. This is driving what's called oxidation reduction potential, and you have to keep your body in the right oxidation reduction state in order for these enzymes to work properly. And what you see is that every single alcohol here requires this cofactor to get it to a sugar and get it out of the body, and this will start to reduce the efficiency of these dehydrogenase enzymes that are utilized elsewhere in the cycle. And so you get a complete inability for the cell, our human cell, to be able to metabolize glucose, fats, and proteins properly through the tricarboxylic acid cycle because it's all being swamped up over here. The result is that you get a cell that looks like it's starving. You'll see an uprise, or in the literature, of what's called non-alcoholic liver disease. It's a misnomer because it's still an alcohol, whether the alcohol is coming from the consumption of foods or the alcohol is being produced in our gut. The cells know no different. They're still receiving a plethora of alcohol and trying to utilize that for energy because it's toxic. And the cell becomes addicted to the alcohol and then is shutting off the ability to be able to use these other nutrients that are vital for our survival. And so the cells look starved. With an excess production of ketones, you'll see an excess amount of triglycerides, cholesterol that might be circulating in the blood. You see altered fat metabolism, sometimes even fat in the stool, which is called steatorrhea. And oftentimes you'll see an increase in lactate, which is the non-oxygen pathway relative to pyruvate. And these are, and there's many other reasons why lactate might be higher than pyruvate, but this is just another, you kind of look at the whole map and what all fits. This is a poor child who is an alcoholic. And when you think about the refined sugars, the simple carbohydrates, the fruits, and where that might be going on in the GI tract, it's not a big mystery to figure out why the cells are looking this way.
Does yeast play a role in that?
Yeast do, and I do get in, so I'm talking about bacteria now, but absolutely. Yeast produce alcohol, that's what we get, the beer from, and yeah. Okay, so it's not a great picture here, so but I just wanted to show you a typical energy, this is the citric acid cycle or the tricarboxylic acid cycle in our human, mitochondria. And here are all the metabolites that you may have learned in biochemistry or when you were in high school and said, okay, I had some sort of acronym to figure all those out and remember the cycle. But every single one of these requires a dehydrogenase enzyme to go through this. Every single one of them requires NAD+ to be in this state for the reaction to be driving forward. So if you're using the dehydrogenase enzymes for produce or metabolizing alcohol, you're shutting down your ability to be able to shuttle through this energy cycle, which is necessary for how fats get in there, how proteins get in there, how glucose is getting in there. There's your question. So that's all focusing on bacteria. And we do have a lot more research on bacteria than we do on any other organism, microorganism in our gut right now. More and more research is being done on the microbiome, which is the fungus. And it's so far believed, and I always have to say so far believed because, it's an evolving sort of story. It's a lot less abundant and diverse, but these fungal populations and bacteria have been together, since the beginning of time, and have an absolute balance and synergy in keeping each other in balance. The Saccharomyces and Malassezia and Candida are thought to be the predominant genre of the microbiome, and with these species being the most prominent. But from the fungal, you can get a lot of mycotoxins, alcohols, aldehydes, oxalates, and these too can start to interfere with human metabolism and produce a lot of inflammatory responses. And just to kind of make sure that we're thinking about this in terms of animals that we consume, phycotoxins, which are part of the Protista kingdom, and that's the classification is very odd, is the flagellated phytoplankton and cyanobacteria, where I never really realized why crab season was shut down sometimes, and why we were getting okay, we can't sell crab this year. I'm like, oh, and for some reason I was just thinking that the crab supply was in poor health or whatever. It's because of domoic acid is being produced by these algae blooms, and domoic acid is an extremely neurotoxic chemical that's being uptaken into these shellfish and mammals and sea lions and whales, and it completely distorts them to the point where you have sea lions, mothers that are just leaving their babies. They're completely neurologically insane. They have no memory of what they even had a pup. They're leaving them stranded, and you're finding marine life having to face a very toxic environment because of the algae blooms. We're hearing about that quite a bit in Florida, and that's going to be absolutely driving a lot of politics with respect to how to control that. This is what drives a lot of the shellfish reaction. So when we think about consuming animals, we also have to think about their microbiome, their environment, and what's going into them. And so kojic acid is actually the toxin associated with the red algae blooms in Florida. So that's microbiome, and even going beyond microbiome into the Protista families. And then viriome. And this is fascinating too, where the viriome is the viruses. And we often think about viruses as being, this virus and vaccine, wipe out. But viruses actually have a beneficial relationship too with the humans, in that they do help with modulating immune responses. And just as we have probiotics on our shelf in the States, it's interesting how the research really has not been funded and got squashed mainly because of politic reasons with the Soviet Union, that the ex-Soviet country, Georgia, very much looks at therapies with viral cocktails as pathogenic to the bacteria. It's a way that these bacteriophage keep bacteria in check by having these various viruses floating around. So they have viral little cocktails on the shelf, like we have probiotics on our shelf, but you never hear of that here. And it's just interesting to kind of think about outside the box approaches to thinking about, okay, it's all a balance and it's all an ecosystem rather than knocking things out. How about putting things in to keep things in check? Clearly, there's also, viral relationships that we have that are not beneficial. And oftentimes, because viruses don't have their own cell wall, they utilize the nutrients from our cell wall to make a capsid that surrounds that virus so that they can evade our immune system. So you see altered, typically, fat metabolism, cholesterol metabolism, when you are infected with particular viruses. And you can then start to see, okay, I'm going to map this out. And, there are, like mycotoxins and things like that not mycotoxins, mycobacteria. I don't know, I'm all of a sudden I just blanked on that. That they also can alter fat metabolism. So it's not just related to the virus. It's also, bacteria have similar sorts of survival mechanisms too. But it's interesting to think about how many viruses actually commonly will alter fat metabolism for their survival benefit on their cell wall, on their capsid. So microbial survival is absolutely driving our behaviors in terms of what we're eating. And so when we think about what we're eating, we have to rewire our whole neurological system. If we're constantly going after this food and we know we feel crappy after that, it's more than just willpower. You're breaking an addiction that's not just our human cells, it's also the microbial cells. And so we have to think about this whole neurological rewiring from the nose into the tongue all the way down through our GI tract and how that's influencing the microbes for rewiring our system. So foods, we can have toxins or nutrients. We've got microbial fermentation, which we have nutrients, byproducts, and toxins that come from that. And then we have our human response to it. That's a very simplified kind of version. What we came up with is a dietary sort of program called Reduced Excitatory Inflammatory Diet, and it's just coincidentally my last name. And, here's my daughter now, and I'll show a picture at the end. She's completely off the autism spectrum as a result of, just thinking about changing the entire way we think about foods and the microbiome within. She was constantly had distended stomach and, gas, constipation, and all of those GI issues just resolved itself with just diet alone. And there's no doubt we did a microbiome turnover in addition to that, although I didn't measure that scientifically because it's very new at that time. But the premise is that we want majority of our foods to be high fiber, nutrient-rich vegetables. This is where the nutrients are. This is the fiber. We're thinking about our human cells. We're thinking about what's going to the microbes. And you have to think about how you're feeding both and getting the best mutual relationship we can. You've got healthy fats and whole proteins. And again, I don't think about proteins in terms of how many grams that I add in my protein powder. I'm thinking, how did I consume that with a whole food? So, for example, like a nut contains, protein, fiber, fats, minerals. I'm getting all of those sources of nutrients from that whole food. And I'm thinking, okay, I've got my protein source, I got fat sources, and I've got, my fiber and minerals. Whole grain fibers, depending on where people are in their journey, can be done. Again, whole grain. We're not talking about, pastas that are processed and stuff. These are things that are looking like the groats that would come off the farm and are not milled or harvested. I like to be able to control my food as much as I possibly can. I trust no one. Except my daughter. She's helping me out in the kitchen lately. And fruits, you think about fruits these days, fruits are just always readily available. Oranges are always in season, but that's not really how we evolved. Fruits should be considered as a treat. And, and thinking about the seasons and how to get variety through the seasons. So the more we eat locally and with our less, small footprint, the more we actually are then driven by some of the variety that comes about from seasons. Now, I like to make sure that I hit this one too. At the top here, we see herbs and spices. And while herbs and spices absolutely flavor our foods and think about that as a a culinary journey in and of itself. It's just amazing how you can take the exact same whole foods and make it taste, more Mexican ethnic one day and Thai another, just based on the spices and herbs that you're adding, and it ends up being a very different, palate pleasure based on all these herbs and spices. It's an incredible way to get variety in also. But what we've lost in our SAD, Standard American Diet, is also its health benefits with really trying to make sure that fermentation is occurring in the right spot in our GI tract. And herbs, and particularly the bitter herbs in that kind of case, we have lost bitter herbs in our dietary approaches. And I noticed a vendor out there whose name has escaped me now, but she's got this Swedish bitters. And I'm like, oh, beautiful, because we need to get these bitters back into our diet to help control where fermentation is occurring. So let me describe that a little bit more. So this is the Reid food pyramid here. So if you look at the GI tract here, ideally we want the majority of fermentation occurring in the large intestine. But that's not where the majority is occurring for a lot of people. A lot of people are having a small intestinal overgrowth of bacteria and fungal, viral narchia, occurring because with a lack of fiber and antibiotics, which is the perfect storm, these microbes are traveling up the lower GI and going into the small intestine and starting to ferment foods there. That's an incredible load of inflammation if you've got fermentation going on in the small intestine. Now you've got, especially any processed food you've got or supplements that you're adding to your diet, you are creating a bloom and perfect environment for a lot of fermentation to occur in the upper GI. And so by adding whole foods and thinking about bitter herbs, you are making it more hostile for these microbes to be fermenting in the small intestine. And these bitter herbs don't impact the lower intestinal fermentation. It's actually helping just the small intestinal balance. And so we don't think about some of that aspect of what we need to be driving is fermentation occurring in the right location within the GI tract. So if you eat a bunch of fiber and you're suddenly noticing that you're bloating within 30 minutes, that's a classic sign you've got upper GI fermentation going on, and that needs to be addressed before you're really going to be able to make great strides with getting healing and inflammation down. And so if we think about when we consume fibers, for example, we are supporting an entire web. We're not just supporting this species, but we've got cross-feeding and, these polysaccharides and insoluble polysaccharides. And you've got this entire web of microbes that are able to be supported with the synergistic activity of all these microbes, relying on the upper layer and the lower layer to support that entire web. And this is critical for getting these short-chain fatty acids in the right proportions through our foods. So short-chain fatty acids, you can find that even the TCA or the citric acid cycle depends on the quality and diversity of the microbiota through short-chain fatty acid production. Okay, and here's just an example of a web that's supported with that. Fiber doesn't come in a bottle. Okay, you've got to get this through the foods, you've got to get this through the nutrients because they require nutrients to ferment also. And if they're not getting it and you just bombard them with fiber, they're going to suck your nutrients right from you. So the fibers, what I like to do is if you have one vegetable for breakfast, see if you can increase it to 3. If you have 4 vegetables for dinner, see if you can increase it to 7. And you're just constantly increasing your amount of vegetables that are going on in the diet. Polyphenols are another thing. These are our colorful vegetables. These are, and herbs and spices. These also support an incredible web of microbial metabolism. Incredible. Huge. So hopefully this empowers you saying, I can control those trillion suckers and I'm going to tell them what to produce and what not to produce. And the age-old saying that we're hearing a lot throughout this whole conference. So you can empower yourself. Tell those little 99% of those suckers, hey, this is what you're doing, and this is how I want to behave. There's my family today. There's my little girl, fully mainstreamed, and every single one of us has observed a health benefit as a result of this journey. And so what I do is I help people, guide them through their journey, whether they want a lot of individual support, hand-holding, recipes, or they just want a little bit of scientific information about how to analyze this metabolic result. We try to help people through that journey. And you can go onto the website, unblindmymind.org, if you want more information, and my contact information is on there. Thank you so much for your attention.